Hi everybody, I'm Fabio. I recently joined the faculty at the University of Padova, Italy. I want to thank the organizers for giving me the opportunity to present some recent work that I've done with a student of mine that is in Denmark, because I was in Denmark until 6 months ago. I want to thank all of you for being here after lunch. Usually it's the toughest talk, so I'm going to try to keep you awake anyway. So if you have questions, just jump in at any moment. Okay, so I'm going to talk about an algorithm that we have developed to find subnetworks that have mutations significantly associated with survival in cancer. But before starting to talk about survival, let me review some of the things that we have already seen in the talks yesterday and also this morning. And in particular, what we've seen is that nowadays we are able to measure somatic mutations in many patients of a given cancer type. And there are at least 2 large projects that are doing that for thousands or tens of thousands of patients. And one of the main goals is to identify driver mutations using all this data. So we want to identify the mutations important for cancer. So one of the ways that is used the most is to use a single gene test to identify such significant mutations. So one can look at each gene one after the other, and for example, counting how many patients it is mutated, compare this number with the probability distribution that is given when we have no driver mutations in the genes. And then by correcting for multiple hypotheses, because we are considering many genes, one can identify a small number of genes that are significantly mutated, and these are the driver genes. As we've heard just before lunch, there are other driver genes that probably appear in the low-frequency mutated genes. And one of the reasons why these genes have low frequency of mutations is because we actually need to look at genes and mutations in the context of networks, since cancer is a disease that is driven by pathways. So our goal actually is to identify the pathways, the set of genes that are significantly mutated. And one approach will be just to look at known pathways, known set of genes, and for each set of gene, counting how many patients it is mutated, and we can just generalize the approach that we use for single genes, use a multiple hypothesis testing on these sets, and we may be able to identify some sets of genes. The problem with this approach is that not all pathways are known, and there are other problems. And so what we want to do actually is to move away from using known pathways and try to identify new set of genes that are important for cancer. So our question is, how can we find significantly mutated set of genes without restricting to known pathways? The main problem is that there are many candidate sets if we want to look at all of them. And in recent years, many methods have been proposed to try to identify important set of genes in cancer without having to essentially look at all the possible candidates. So while I was compiling the list of all the methods, I saw that actually couldn't fit all of them in the slide. So what I've decided to do, taking inspiration from a speaker from yesterday, is to just decide to not put any of the previous methods here and just talk about 2 classes of methods that I've contributed to. The first one is the class of methods that actually have been presented by Max just before lunch, that look for patterns of exclusivity among mutations. And the second class of methods are methods that try to combine an interaction network with mutations to identify the significantly mutated subnetworks. And I've contributed to this with the method called HatNet, and with this improvement, HatNet2, that is joint work with Max Leiserson. OK, so why did I present this? Because I want to make the point that by moving from single gene test to actually looking at a set of genes, we can identify low-frequency genes or genes that may have a low score by themselves. And by pulling them together in sets, we may be able to identify more genes that are significant for cancer. OK, so in many studies, for example, from the TCGA, in addition to the mutation data, we also have survival information. So for each patient, we are given a survival time. And so a natural question, when we have this data, is try to find mutations that are associated with survival. So what I mean is that we want to identify, for example, mutations or genes such that if I compare the survival of patients that have a mutation in the gene with the survival of the patients that do not have a mutation in the gene, the survival is different. So how can I do that? Well, let's look a bit more closely to what is actually the survival data that we get. And the survival data is actually made of two pieces of information for each patient. One is the survival time, the time to the event for the patient. And the second piece of information is the censoring information that tell us if the survival time that we have for the patient is the actual time of the event, for example, of the death of the patient, or is just a lower bound for the patient. In that case, we say that we have a censored survival, censored information. So now, given the mutation data and the survival information, how can we identify the mutations or the genes that are significantly associated with survival? Well, by analogy with the case of finding driver genes in cancer, we can start from single gene tests. So I can look at one specific gene and partition the patients into the two groups, the patients that do not have a mutation in the gene and the patients that have a mutation in the gene. I can represent this with a group label, essentially. And now this splits the patients into the two groups, compare the survival distribution that I can estimate quite easily, and then use a statistical test to obtain a p-value that is a measure of the difference in survival between the two populations. And this essentially is a measure of the association of the mutation with survival. OK, so what is the statistical test that we are considering? One of the most used statistical tests is the log-rank test. Oops. Is the log-rank test. That is a non-parametric test in the sense that it doesn't actually consider the actual times, the observed times of survival. So what we do is just we consider the patients sorted by increasing survival time. And then all the information we need to perform a log-rank test is the group label, so to know if a patient has a mutation in the genes or not, and the censoring vector. So actually, since the censoring is fixed for the entire data set, and now we will look at different genes, what changes is just the label information. So the vector X of the group labels. And what the log-rank does, it just computes a test statistic. So it's just a function of the data that depends on the group information and the censoring. But as I said, we can consider it just as a function of the group information. So we can look at this. For a given gene, we can compute a score. And this score just depends on the group label. So the log-rank test, to not go into many details or any more details, give us a way to compute the probability that the statistic that we observe in the data comes from or observing a value for the statistic at least as extreme as the one that we have in the data under the null hypothesis of no difference in survival between the two populations. So the smaller this probability it is, the more unlikely it is that the difference in survival that we observe for the two populations is actually explained by random effects. OK, so now that we have the log-rank test, what we can do is just perform a single gene test for the first gene, a single gene test for the second gene, a single gene test for the third gene, and so on for all the genes. We obtain a p-value different for each of them. Then we apply multiple hypothesis correction, and we'll obtain a list that tells us which ones of the genes are significantly associated with survival and which ones are not. So let me add a point to this. This seems all very easy. This approach actually requires the accurate computation of small probabilities. And so you cannot actually use standard tools for survival analysis that are based on the asymptotic approximation or asymptotic distribution for the log-rank statistic. So you need to do something different from what is, for example, implemented in RSARVDiff, just to mention a specific software. So what we've done in the past is we have developed a tool that provides an accurate approximation of these very small probabilities that are needed in case you are testing all possible genes in the genome. OK, so now that we have this test, what we can do is take a real data set, for example, from the TCGA Glioblastoma Multiforme, which we have hundreds of samples. We take the samples for which we have mutation, survival data. We take only the 780 genes that are mutated in at least 3 samples. And we do this single gene test. What we obtain out of this is actually that there are no genes that are significantly associated with survival after the multiple hypothesis correction. So this is actually a common picture when you look at the dataset from the Cancer Genome Atlas. Usually you obtain one or let's say few genes associated with survival, or very often you don't obtain any gene associated with survival. So why is that? Well, it might be that there are no mutations or genes associated with survival, but the one possibility is actually that we need to look at set of genes and try to understand if the set of genes is associated with survival. So what do I mean with this? Is that, for example, if I look at 2 genes, let's say even they are interacting genes on a network, for example, what I can do, I can partition the patients based on the mutation status for these 2 genes. So I can divide the patients into the ones that have at least one of the 2 genes mutated and the patients that do not have any mutation in the genes. And now again, I can use the log rank test to measure the difference in survival between these 2 populations. Okay, this is one when I'm given the set of genes. What if I want to find the subnetwork that is associated with survival? So I'm not given the set beforehand, but I wanted to find it. So maybe I want to identify the set of genes among all possible genes, or maybe what I want to do is to restrict to a gene interaction network. So the problem that I want to solve is, given the mutation data, the survival information, and this large gene interaction network that contains tens of thousands of genes, I want to find the connected subnetwork, restricting maybe to k genes, that has the highest association with survival, as measured by the log rank test. So this is a computational problem. It's not well formulated yet, because I haven't said what it means to have the highest association with survival. So the way we can measure this association through the log rank statistic is by defining for a set of genes its weight as the normalized log rank statistic that I obtained by the group label I described before. So I can take the patient, the set S of genes, and assign the group label according to a procedure I've said before. So I assign a 0 if I don't have any mutation in the set of genes for the patient. I assign a 1 if at least one of the genes is mutated. Now that I have the group label, I can compute the score for this group label, and that is going to be the score for my set of genes. Okay, so now I can really define a computational problem that we call the max connected k-set log rank problem. And it corresponds to identify the subnetwork of k genes of a large graph of n genes that maximizes the log rank statistic, or this score. So the first result we had in a paper that is going to appear in Recomb 2016 is that this problem is NPR'd. It is NPR'd even when we restrict to graphs that have only one node with high degree. And since in biological networks there are hubs, this problem is NPR'd to solve in practice for networks of biological relevance. Okay, so there are many problems that are NPR'd on graphs in bioinformatics, but they are solved anyway. And during these years, many techniques have been used to solve graph problems. And in particular, a technique that it was to interest to us is the color coding technique that has been introduced by Alon, Yuster, and Zwick in STOC 94. So how does the color coding technique works? Let's say that I have a graph, a network, and I want to identify a subnetwork of size 3. What I do is I random color the graph with k colors. So in particular, I assign to each vertex in this graph a random color chosen among 3. And why is this a good idea? Because once I have a given coloring, finding the highest scoring colorful subgraph is easy. A colorful subgraph is a subgraph that contains all k different colors, and it is connected. So for example, this subgraph up here is a colorful subgraph with 3 genes. So easy means that it can be done by dynamic programming, and this procedure, this dynamic programming procedure, is efficient as long as k is not too large. So as it is essentially log n, where n is the number of vertices in the entire graph. Yeah?
So like, where's the score?
What is the score? Great question. So the score for us is going to be the log rank statistic.
So here in this problem, they also had associated scores for subsets.
In general, no. In general, it has been applied to many different scenarios that I'm going to present in the next slide, just to mention. The score can be different things. For example, you can have a single gene score that represents some measure for a gene. You want to maximize maybe the sum of these scores. Or you may just be interested in finding certain structures. So you actually don't have a score for the gene. You want to maybe understand if there is a simple path of a certain length k. And so you are looking for a colorful subgraph that is a simple path. But the main point is that you can find, let's say, high scoring or colorful subgraphs that have certain properties efficiently when k is not too large. Okay?
But just to follow up on that, so
yeah.
For dynamic programming to be used here, you cannot have any score, right? You need the score to be additive or...
Great point that I'm going to mention later. Yeah, you need the score to be additive. So hold on that for 2 more slides, I think. And then I'm going to go back to that. OK. So in this approach, now we have obtained one random coloring of the graph. And the problem is that maybe your highest scoring subgraph, like the one that has the highest association with survival, may not be colorful in the coloring you have chosen, but it's easy to see that if you repeat this enough times, then actually in at least one iteration, your subgraph will be colorful. OK, color coding has been used a lot in bioinformatics for many different applications. And in particular, I think it has been used at least by Roded, Cenk, Iman, and Ron. There are many of the people inside in this room that have used color coding. And so what we have done with my student Tommy Hansen, that is currently at the University of Southern Denmark, is to build on top of the color coding procedure a method that identifies the subnetworks with mutations associated with survival by taking a gene interaction network, run a diffusion process on the network so that we capture interactions that are not just direct interaction. And then we combine this network with mutation data and survival information. And using the color coding, a color coding based algorithm, we identify subnetworks that are high scoring, where high scoring means they have a high normalized log rank statistic. Once we have identified these subnetworks, we know that they have a high log rank statistic. So it means they will show a significant difference probably in survival between the patients that have a mutation in the subnetwork and the patients that do not have a mutation in the subnetwork. However, we need to perform a permutation test to make sure that what we are finding is not just an effect of random fluctuations in the graph. And I can talk more about that if you have questions about that. OK, as pointed out by JP, when I before said that given a coloring of the graph, finding the best colorful subgraph is easy, I was actually cheating in the sense that this is true only when the score is additive. So only when the score for a set of genes is given by the sum of the scores of the single genes. So what does this imply for our problem? Well, our score is not additive. That means that color coding may not find the optimal solution. Actually, in the recon paper, we have the proof that with the addition of few vertices, in the worst case, we will not identify the optimal solution. So why does it make sense to use color coding to solve our problem? Because the data that we get is not worst case. And so what we proved is that under certain assumptions for mutations and their relation with survival that we think are reasonable, and mostly they mean that most of the mutations are placed independently of survival. Under this assumption, if you are given enough samples, then our algorithm NOMAS will identify the optimal solution. So we'll actually identify the subnetwork that has the highest association with survival. Okay. So Tommy has implemented this algorithm, and we tested it on data, first of all on simulated data, and then on cancer data from the TCGA. So on simulated data, what we've done is we assume that there is a set D of genes that have association with survival. The set is not too large. In particular, for this experiment, I think it was around 10. And what we have is that each of the genes is mutated with a certain probability P in a set of patients that have low survival. But instead, for all genes, they also have random mutations that represent passenger mutations or mutations that are independent of survival. And then we run NOMAS and compare the solution, the best solution found by NOMAS, with the subnetwork D that we planted in the data. And so what we are showing in this graph is the probability that the solution found by NOMAS corresponds to the planted solution as a function of the number of samples that we are using in the data. So as we can see, for example, for this red line, that corresponds to the case of when there is not a strong association between the set D and survival, the probability that our algorithm identifies the subnetwork associated with survival is not that high. But when the number of samples increases, or when the association between the set and survival is higher, our algorithm finds the actual planted solution with probability that is getting closer to 1. And moreover, simulations show that even when we do not identify the optimal solution, the solution we find contains many of the genes that are actually associated with survival. And since we are using color coding, actually, we don't have only to look at the highest scoring solution. We obtain other solutions for free from this dynamic programming table. And so what we have is that if we look, for example, in the top 10 solutions identified, we identify the highest scoring solution with a probability that is at least 50% for our for sample sizes that are around what we have for glioblastoma multiforme, and with higher probability when we have more samples. Okay, so then we tested NOMAS on cancer data from the Cancer Genome Atlas, and we used the network that we used in the HATNet 2 paper as well. And we compared in particular NOMAS with other approaches. In particular, we compared with greedy algorithms that have been used before to identify subnetworks associated with survival. And these are actually greedy algorithms that are inspired by a standard strategy, a standard greedy method on networks. And also we compared with methods in which the score now is just an additive score on the genes. So this is essentially an approximation of the score. So what we have when we run NOMAS is that if we look at a specific data set, for example, and these are the two tails of the log rank, we can focus on one of them, the score of the solution, the top solution identified by NOMAS is represented here in black, while the solution found by the other methods are represented with the other lines. And we can see that NOMAS always identifies better solutions. Sometimes the other methods identify solutions that are quite close, but sometimes they are actually very different. So actually our method does identify better solutions on real data. Okay, now in the few minutes that I have left, I'm going to just show you two subnetworks that are identified one on glioblastoma multiforme and another one in AML. And this is to be considered essentially work in progress. So this is a subnetwork that has association with low survival, in the sense that if you look at the patients that have at least a mutation in this network, the survival distribution for these patients is lower than the survival distribution of the patients with no mutation in this subnetwork. And this is a representation of the mutations and the different genes in the network. You can actually see that since the patients are sorted by increasing survival time, most of the mutations in the subnetwork appear for patients that have low survival time. So this is significant according to a permutational p-value, so it is highly unlikely that you would identify such a set of genes in a network. And The most important thing is that, well, first, one of the genes, CDKN2A, is a gene that is known to be important for glioblastoma multiforme. And none of the genes has significant association with survival when considered in isolation, as we said before. So the entire set is significantly associated with survival, while none of the single genes is. Then, as I said, this is work in progress. So the figure that I have now for AML is not as nice as the one that I had before. But this is a subnetwork with 8 genes identified on AML. Again, none of the genes has significant association with survival, and the p-value is still significant for the entire subnetwork. This subnetwork shows mutations that are associated with increased survival. And in fact, what we have done in this data set, we have also included fusion genes that are important for AML. And there are PML and MYH11 in this subnetwork are actually fusion genes that have been reported to be associated with favorable cytogenetic risk in AML. Okay, so with this I conclude, and I want to just thank the collaborators at Brown that have been involved in the methods that I've talked at the beginning, Ben Rafael, Eli Upfa, Max Leiserson, and Shinta Wu. And I want also to acknowledge my student Tommy Hansen, that is the one that has done most of the work for the RECOM 2016 paper. Thank you.
Questions? So you skipped over the part about censoring, but isn't it true that if you knew the true survival time of the censored patients, that would alter the ordering, and that's alter your log rank? Do you know how significant that might be?
I'm not sure how significant that might be. What I can tell you is that actually the log rank and the test for the statistical test for survival analysis have been designed so to take care of the fact that there is censoring, and censoring is affecting a large proportion of the patients. And one of the assumptions that is made by this method is that censoring is essentially independent of the survival time, so that what you get is, the two things are not correlated, and so the statistic, or you can obtain a nice distribution asymptotically.
Simple example, you compare against the censored data or the combination of both, the uncensored data versus both, you get roughly the same result?
Well, no, the two things I think may be different. I'm not sure. I haven't tried that. But it may be something interesting to try.
I had a question about your connectivity constraints. So if you go back 2 slides to the module that you showed. Yeah, this one, for instance. Yep. So ubiquity, UBC has a super high degree, so it's kind of like a hub. And I was wondering whether those tend to be more overrepresented in your solutions.
They don't tend to be overrepresented in our solution, so we don't get all hubs in here. And that's, but you are right that that is a hub. It has degree, I think in our diffuse network, it has degree probably 400 something. And that's why we use the permutational p-value, to try to make sure that what we are getting is just not a random set of genes around a high degree node.
And a follow-up question, quick one. So if you look at, if you do the color coding, your randomized algorithm, so the suboptimal solutions, what do they look like? Are they much different, or do they? So the p-values, if you just look at the p-values, so like.
That's a great.
Or do they overlap a lot? Or?
That's a great question. They tend to overlap a lot in the top ones. What I'm showing here are just the top ones, and we have actually. It's easy to extend the permutation test to take care of the entire distribution, but that's a great question. You can get many solutions that are actually very similar. And as I said, this is work in progress, and one of the things. Can you summarize all the solutions that you get?
So I'm sorry, I didn't really get what the score is. So you said it has to be additive. So is that the sum of the
okay.
log rank statistics of the?
No. So the score is not additive. Our score is the log rank statistic, the normalized version, that is not additive at all.
Okay.
So in the worst case, the color coding does not identify the optimal solution. Okay, so if you get worst case data, you can, or let's say differently, you can build an example where using color coding you will not get the optimal solution. But what we prove is that if the data comes from a distribution that we think is reasonable, then actually if you have enough samples, what you will get out of the color coding technique is the optimal solution.
But you still run dynamic programming?
Yes, it's a dynamic programming, and there are a few things that we do in the color coding part to speed up a bit compared to other, to the standard application of color coding. I can talk more about that with you after.
Maybe very last step.
So I understand that with this with this requirement that you want to have a connected subgraph in the protein-protein interaction network, you have a very nice computational problem. You can use very nice solutions and so on. But I was wondering about the biological background, because I mean you're not looking for a mutually exclusive pathway of genes. So what is the actual reason why you want to have these genes connected? Because they might be in completely different pathways. They are completely away, and they are then associated with survival.
Yeah, so that's a great point. I guess the motivation here was to try to look for pathways, so that the entire pathway is associated with survival. So it might be that you have a mechanism essentially that is associated with survival, not a single genes in there. But actually one of the directions we are looking at is to find genes that may be in different parts of the network and still be associated with survival, because it might be that not all the genes in the pathways are actually have the same effect on survival, let's say. So you're absolutely right. This was this is an attempt to trying to find pathways or group of interacting genes that are associated with survival.
I think this is a great discussion topic too. So not just for this, but in many of these papers that, and I've been a culprit myself, why do we insist on connectivity? In what way do we identify a pathway? Maybe one day we should question the assumptions that we have.
I was thinking that maybe the discussion this afternoon could be about networks, because
yeah, that sounds good.
We heard Igor's talk this morning, and we've now seen more network stuff. There'll be more talks on networks, but sort of what are the dangers of using various networks? What do we know? What do we don't know? What are the assumptions that go into the pathway analysis? That would be one sort of topic.
And let me mention. Let me just mention, even if you take the network away, it's still NP-hard.
Of course.
Very difficult.
Sort of related just to this. I don't know how much treatment information there is, but I wonder if that having treatment annotations on top of that would actually pull out differential survival, because now you're kind of associating the genetics to.
Yeah, that's a great point. So I guess a general question is if you have phenotypic annotation or different type of annotations about the therapy, can you do a better job? And for some of the data sets, at least, there is such annotation. So I think we can do a better job, but it's an open direction, essentially.
Let's thank Fabio again.